All right, we're going to talk again about how cells divide. We've got to go back into chapter 6 and pick up a few things that we kind of skipped over, but have some importance. So we're going to describe cell division in prokaryotes like bacteria and archaea. And first I'll describe their chromosomes a little bit, and then I'll talk about the process, which is called binary fission. And then I'll show you how it relates to mitochondria and chloroplasts and talk about the evidence for endosymbiosis. So um, if you recall, I talked about eukaryotes having a lot of DNA. So that means obviously prokaryotes have a lot less. They only have one chromosome. It's usually a circular chromosome. And for like E. coli, they only got around 5 million base pairs. That's more than 1,000 times less than humans have. And so much, much less DNA, and it's on a single circular molecule. So here, cell division doesn't need to be so complicated, and it isn't. They use a process called binary fission, which begins with the replication of the DNA molecules. And here there's an origin of replication where uh, the enzymes grab hold of the DNA and start copying it in both directions. And so this origin of replication, at this point, you start getting a bubble, and it's called the rolling circle method of replication. And the first thing that's replicated are these origins. And so they will be the farthest apart of any of the molecules, and they'll be later become anchored to the cell wall. And so at kind of opposite ends of the cell. And so what'll happen is once replication is pretty much complete, you'll have a protein called the FITZ or FTSZ will start constricting the cell at between the two chromosomes, and it'll form a ring and it starts constricting the cell, and a septum forms at this part where you get new cell wall, and eventually you'll have two cells. Um, this is simpler, faster than what you find in eukaryotes. Okay, and it's all because you only have one chromosome. As long as you can form the division between the two replicated chromosomes, this is pretty easily. And so, like in E. coli, because this is so easy, it can happen relatively fast. And so here I have two pictures of E. coli, one showing the whole cells. The DNA is stained red here. The septum is forming between them and it's pinching. Here you just see the septum kind of at the beginning, and this only takes about 20 minutes. So it's really kind of nice that it happens so fast. And we see a similar process happening in eukaryotic organelles, not all of them, but some of them. In chloroplasts, we see an almost identical process following DNA replication. The chloroplast chromosome, again, is a single circular chromosome, like in bacteria. It's much smaller, only about 120,000 base pairs. Um, and but the replication process is very similar. There's no prophase, metaphase, anaphase. What happens is, again, you have an origin of replication, the rolling circle method. Their two chromosomes become anchored far apart, and you get a constricting ring of protein. In some cells, it's got the FTSZ protein. Others have primarily the plastin dividing rings and dynamin rings. So kind of shown here, they show the PD protein, that's the plastin dividing protein. And again, you get a constriction forming, and this is how these organelles reproduce. And it's very similar to the reproduction you would find in cyanobacteria, and those are the blue-green algae, and they share gene sequences with the blue-green algae. Likewise, you have a similar process in mitochondria. They have their binary fission again following DNA replication. Again, here the chromosome is quite small. In humans, there's only about 10,000 base pairs on that single circular DNA chromosome in the mitochondria. But again, it involves several proteins that form a constricting ring between the two chromosomes. Um, in humans, the protein is DRP1, which forms a ring, which is a cytoskeleton related protein. But in some primitive eukaryotes, they do have the FTSZ protein, particularly in single-celled eukaryotes called protista. And again, this is very similar to a particular group of bacteria. These are aerobic organisms in the alpha proteobacter group. And so here I show kind of the upper picture here is mitochondria in the process of having that constriction and dividing, and the lower one is the diagram. So here what we think is that we've got a good piece of evidence for that endosymbiotic origin of mitochondria and chloroplasts. And I should say there's a whole lot of evidence for this. These organelles have double membranes. There are single circular chromosomes in these organisms, just like bacteria. They undergo binary fission. Their chromosomes use a 70S ribosome to be expressed. And again, we can identify gene sequences. So here what we think happened is the ancestral host cell uh, took in a mitochondria, and then in some organisms, it took in a chloroplast. And so that's how you got plants, a photosynthetic algae. We also think that we can identify the ancestral organisms not only for the chloroplast and mitochondria, but also for the nucleus. Okay, we have found groups of archaea that show a lot of similarities to eukaryotes, that these groups have cytoskeletons that contain both actin and tubulin genes, not found in most prokaryotes, but found in these guys. They also have histone proteins for winding DNA. Um, they do contain binary fission, but we also know of archaea that have linear chromosomes. So this may be uh, linked to the beginning of mitosis. And of course, we have gene sequences that are common, and ADS ribosomes are similar, more similar to the ribosomes in archaea than they are in bacteria. So we think these Asgard archaeans are ancestors to the nucleus. Okay. Here, all we need to kind of go is think back. Okay, we're in a group with the archaeans where some of them had multiple chromosomes and they were linear. Not many archaeans have that, but there are some. Not the Asgards, but relatives that aren't too distant. Okay, so that takes us back to eukaryotic chromosomes, lots of DNA, much more DNA, many associated proteins. Some of those proteins we find in archaea. These linear molecules are also somewhat found in archaea, and the central role of the cytoskeleton kind of suggests that one of the ancestors of eukaryotes must have had cytoskeleton proteins, and we can identify the group of prokaryotes that had these. They are the Asgard archaeans. So kind of brings together some of the evidence for the origin of eukaryotic cells, and identifies some of the potential ancestors. So kind of cool stuff. All right, I'll see you on Wednesday.